Welcome to the Cell-Cell Communication and Development module. In this talk, we're going to learn about Hedgehog signaling. Paracrine factors are diffusible molecules made by cells that can act as inducers or ligands. They work in a short range of 40 to 200 micrometers, which is roughly 15 cell diameters. Paracrine factors can mediate inductive events through the interactions that are called as paracrine interactions. Now, autocrine interactions are interactions when a cell that makes a paracrine factor is also able to respond to that factor. We normally do not observe autocrine interactions during the processes of development. Now, morphogens are paracrine factors that are able to mediate different developmental processes by forming a gradient. Morphogens are able to determine the fate of a cell by their concentrations.
Thus, just to summarize, morphogens are secreted signaling molecules that affect gene expression differently at different concentrations. Uncommitted cells that are exposed to high concentrations of the morphogen are specified as one cell type, as shown in the figure here. So here we see that there's a certain concentration of the morphogen that allows the cells to become committed to form a specific type of cell. Now, when the morphogen's concentrations drops below a certain threshold, then the cells that are experiencing that lower concentration, they become different types of cells, or their fate is normally altered. Finally, if the concentration falls even lower, then a cell can be specified to develop into a different type of cell altogether. Thus, the morphogens affect development based on their concentrations. Now, the range of a paracrine factor or morphogen depends on the synthesis of the factor, so how much is being made by the cell, the transport of the factor, so how much is being transported to the cell's surface and secreted out, and finally the degradation of the factor, so how stable is the factor and how easily is it degraded. Now, paracrine factors that play important roles in development can actually be grouped into four families based on their structure. They are the fibroblast growth factor family, or the FGF family, the Hedgehog family, the Wnt family, and finally a superfamily called as the TGF-beta superfamily. The TGF-beta superfamily comprises of many different types of groups, which includes the TGF-beta family, the activin family, the bone morphogenetic protein or BMP family, nodal proteins, etc. Now let's look at the basic mechanisms of signal transduction. So normally the cells have receptors that will be able to associate with the ligand, which is the paracrine factor. These receptors can have intracellular enzymatic activity as shown in the proteins that are present in the blue box. On the extracellular side of the cell, they will have a ligand binding domain. Now, the ligand, which is the paracrine factor, can bind to this receptor, and normally it causes conformational changes in the receptor, which results in activation of the intracellular part. So when we look at the intracellular part of the receptor, the binding of the ligand triggers a conformational change that can activate the intrinsic enzymatic activity of the receptor. If the receptor's intrinsic enzymatic activity is that of a kinase, then they normally phosphorylate each other to become functionally active. This activated receptor can now perform a function. So in this case, since it is a kinase, it can actually add a phosphate group to a protein. Now it so happens that this protein that's present in the cell is inactive when it is not phosphorylated. However, upon phosphorylation, it becomes active. And now this phosphorylated protein can relay the signal by activating other kinds of proteins. The next family of paracrine factors that we're going to be looking at is the Hedgehog family. These are multifunctional signaling proteins that act in the embryo through signal transduction pathways and were originally found in Drosophila. Three homologs of this family are found in vertebrates. The first one is called Sonic Hedgehog, in memory of the cartoon character Sonic Hedgehog, and it plays important roles in many aspects of development, including motor neuron and vertebrate development, hair or feather formation, etc. Another homolog is called Desert Hedgehog, which plays important roles in spermatogenesis. The third homolog is Indian hedgehog that plays important roles in postnatal bone growth. Hedgehog is a morphogen, and different modes of processing and assembly can significantly alter the amount of hedgehog that is secreted and affect the gradient that is formed. Hedgehog can also associate with other molecules like cholesterol and palmitic acid, which can influence the gradient that is formed. So when we look at the formation of hedgehog, hedgehog is translated in the cytoplasm. However, since it has to be secreted out, the ribosomes will pause when the signal peptide is formed and then will dock to the endoplasmic reticulum membranes and continue translation. So that is what is shown in the blue box, where we can see that the translation of hedgehog is completed in such a way that the protein is being threaded into the endoplasmic reticulum. Once in the endoplasmic reticulum, once hedgehog forms its proper conformation, it undergoes autoproteolytic cleavage. This autoproteolytic cleavage part can now be associated with molecules like cholesterol and palmitic acid. It will then be transported from the ER into the part of the plasma membrane. Now, there are four ways in which it can be secreted. The first way is it can be secreted in the form of monomers. The second way is it can be secreted in the form of multimers, where we can see many hedgehog molecules coming together to form a multimer. They can also associate with lipoproteins and form a lipoprotein assembly. In these cases, we can also see ECM interacting proteins like heparin sulfate proteoglycans. It can also form these exocytosis vesicles and be secreted that way. Thus, there are many modes in which hedgehog can be secreted from the cell. Now let's look at the signaling pathway that is induced by hedgehog. The hedgehog pathway is similar in Drosophila as well as vertebrates. What we're going to look at in the figure is the hedgehog pathway in Drosophila. So in the cells, there's a receptor called patched, which is the receptor that will be binding to hedgehog. Now, in the absence of hedgehog, patched remains on the cell membrane, and it inhibits a specific protein called smoothened and promotes its degradation. The degradation of smoothened results in the phosphorylation of a protein called Ci, and when Ci is phosphorylated, it can get cleaved into two portions, and one of the cleaved portions can bind to DNA and act as a repressor, and this way we do not see gene expression. When hedgehog is present, hedgehog will bind to patched, and hedgehog binding to patched promotes the degradation of patched. Since patch is being degraded, smoothened becomes stabilized, and now smoothened in its stabilized form can get phosphorylated. The phosphorylated smoothened can now cause the Ci protein to not be phosphorylated because the kinase that phosphorylates Ci is now no longer in close proximity. This unphosphorylated form of Ci can bind to the DNA, and in this form, the CI is actually an activator of gene transcription. Thus, Hedgehog is able to activate genes. During development, the Hedgehog pathway is extremely important in many processes. This can be observed in the figure in the blue box where we can see the expression of hedgehog in an embryo. Wherever we see the shades of blue or purple is where hedgehog is there, and as we can see in the figure, it is in many places in the embryo. Some of the processes that hedgehog plays a role in is vertebrate limb patterning, neural differentiation and pathfinding, retinal development, pancreas development, craniofacial morphogenesis. Now, mutants of sonic hedgehog have been shown to have major limb and facial abnormalities, thereby suggesting its important roles in limb and facial development. An example is when we have mutations of sonic hedgehog, we can see cyclopia being formed or shown. In the case of cyclopia, where we can see in the figure shown in the blue box, the organisms have only one eye and they have many different types of facial abnormalities. Hence, hedgehog plays an important role in development.
With this, we come to an end of our talk where we learned about paracrine factors and morphogens, as well as different factors that can affect the range of these paracrine factors and morphogens, as well as different types of morphogens that play a role in development. We then concentrated on the hedgehog signaling pathway.